Hi there, welcome back to Chapter 19, Blood with Professor Howard, and we are moving on to white blood cells, hemostasis, and beyond. So hopefully during this video we'll finish the blood chapter and I'll then end this video and start working on the next one. As I mentioned, these take a long time to film, so this stuff takes time. Alright, let's do the thing. So, we talked about red blood cells, how they're made, what's on their surfaces, details about typing, and now we're moving on to white. Fortunately, since this is asynchronous, I won't have to field the same questions that I usually do during this lecture. And what happens is students have a lot of questions about the specific functions of how these cells work, and I usually have to say something like, hey, we're not going to do that right now, we'll get to that when we get to the immune system chapter. But now I've just said that. So this is going to be sort of a very cursory examination of the white blood cells. We're not doing much that's super specific, and that's for a reason, because they have their own chapter where they are the stars of the show. So if it seems like I'm going through these pretty quickly, I'm doing that on purpose because we're going to spend a lot of time on these guys a little bit later. So as you guys know from lab, you've got your granulocytes and agranulocytes. So these are your granulocytes, and these two are your agranulocytes. Generally speaking, if you see white blood cells in the bloodstream, say in a blood smear on a microscope slide, that means they're on their way somewhere. So the ones that you see in blood are in transit. That's not where they spend the majority of their time. So they roam around in loose and dense connective tissue. And they generally are attracted to chemicals released from damaged tissue. So things like prostaglandins and other substances that are cell signalers. So what they'll do is they'll leave the bloodstream where they sense these and other eicosanoids and go to that place where they're needed. So the chemical cue for them to arrive are the cell signaling molecules indicative of damage. So another name for white blood cells is leukocytes. And unlike red blood cells, these are nucleated. They have a variable lifespan, so it really just depends. We're not going to discuss that in detail here. They do not carry hemoglobin and therefore have no oxygen carrying capacity. They certainly use oxygen, but they don't carry it around. And these are your defense force. So between all of the different kinds, they can protect you against an enormous variety of different pathogens, including pathogens that are living, like bacteria and parasites, and pathogens that are non-living, like toxins, venoms, and technically viruses are not alive either. So I mentioned granulocytes versus agranulocytes. You guys know what that looks like, right? Because you've seen those granules on the lab test. And compared with red blood cells that are super numerous, there's between 250 and 280 million per red blood cell. These are only 60 to 90,000 per microliter, so that's a very, very small amount. And that's reflected in the relative infrequency of them observable on a blood smear. So the process of them migrating out of the blood is called diapedesis. And before that, they do something called margination, which is just a fancy way to say ordinarily these cells, as they're traveling in blood vessels, avoid the vessels they're traveling in, but during margination they stick to the wall. And they move sort of like amoebas, so they're going to extrude their cytoskeleton inside of their membrane and sort of push their leading edge out. And then we discussed this already, so they are attracted to damaged tissue signals, as well as cytokines from other white blood cells. So these guys are going to release, which are a class of chemicals that basically are ways that cells talk to each other. So positive chemotaxis means following a trail of chemicals from an area of lower density to an area of higher density. So you're going up the smell trail to the source of the smell. And then in some cases, we have cells that are going to be avidly phagocytic, and these include monocytes, eosinophils, and neutrophils, which is what MEN stands for. Okay, So neutrophils, another way to say them is polymorphonuclear leukocytes, but that's obviously a huge mouthful, so we're just going to call them neutrophils. As you know, they're the most numerous of the white blood cells, and these are bacterial specialists, so they really like to attack and kill bacteria specifically. They're capable of doing other stuff as well, but they're generally the first on the scene in places where bacteria have managed to gain access to the body somehow, usually by a cut or a scrape on the skin. So the granules that are appearing in their cytoplasm contain lysozyme and other bactericidal compounds that are designed to specifically attack bacterial cell walls and membranes. And these guys are also really, really quick. So because they're relatively small, they perform margination and diapedesis pretty snappily, and they're usually the first white blood cells to arrive. So these guys secrete leukotrienes, which are another class of chemicals. Leuko means white, and trienes, think of that being like triage, right? So they're going to triangulate and triage more leukocytes to come to the place where they're needed. And of course, I already mentioned prostaglandins. So neutrophils have a relatively short lifespan. After they differentiate, they only exist for about 10 hours, which means, again, that although they are the most numerous, that means that your bone marrow and your hemopoietic tissues have to be constantly making neutrophils. So they don't end up being that old. And that's in big contrast to some of the other white blood cells that can be much older than that. Finally, before we move on, I just want to point out that these guys are capable of basically laying waste to bacteria. So they can take care of and kill up to 24, so 24-ish bacteria in less than 30 minutes. So they're very good at their jobs. Eosinophils, remember these are the second rarest. Never let monkeys eat eosinophil, banana, basophil. So these are only 2 to 4%. And they have this big bilobe nucleus and reddish-orange granules. And those granules that you guys learned to identify the eosinophils by are little granules of toxins. So basically, the eosinophil arrives at some kind of target that is coated with antibodies. It's going to barf up a bunch of toxins onto that targeted antibody-coated thing, whatever it is. And then once the target has been disabled, then the eosinophil will proceed with phagocytosis. So this includes bacteria as well, although that's not their primary goal. They're also heavily involved in antiparasitism, so they can attack blood-borne or tissue-borne parasites, and they help clean up cellular debris. So obviously, neutrophils are mostly doing the bacteria, and macrophages are mostly doing the cellular debris. But eosinophils help with that task. Their speciality, however, is protozoa and parasites. As you know, basophils are quite rare, and here you can see a classic basophil appearance. So dark granules, a nucleus that is bilobed, but you can't really see that usually. And in the case of basophils, the granules are made of histamine and heparin. So these are going to locally thin the blood and dilate the blood vessels to allow other white blood cells to have an easier time getting to them, basically. This is the same thing that mast cells do. Basophils are just giving the mast cells in the connective tissue an assist. Okay, monocytes. So monocytes make up between 2 and 8% of white blood cells, and they move into tissues within 24 hours of maturation. So again, I've characterized monocytes before as being baby macrophages. That is true. So the ones that you see in blood smears have recently been formed within the last 24 hours. And so they really are baby macrophages. They're very, very new when you see them in the blood, and they're on their way to wherever they're going. So they spend a very short time in the bloodstream in that stage, which partially accounts for their relative rarity. So only 2 to 8%. So these guys release chemicals that attract both phagocytes and fibrocytes. So another way to say this word is fibroblast. So remember, these guys are attracted to damaged tissue, and they're going to clean up cellular debris and other damaged stuff. And if you have damaged tissue, that implies that you're going to need to replace it, right? So it makes sense that these cells would attract fibrocytes as well to start filling in the gaps where the damaged tissue used to be. So after the monocytes migrate out of the tissue, they become macrophages. And you can broadly divide these into two categories. So the fixed macrophages basically stay put. It doesn't necessarily mean they stay put in one place forever, but rather they are sort of going to stick to the organ that they end up in. So if it's not clear what I mean is it's not like they become a rock where they just stay in one place in the organ forever and ever and ever. But if it's a macrophage, for example, that lives in the lung, it'll crawl around the inside of the surface of the lung, but it's not going to leave the lung. So that's what I mean when I say fixed. That doesn't mean they're immobile. It just means they stay in one place, that place being the organ. So fixed macrophages are really important for tissue health. So for example, the lungs, fixed macrophages are also sometimes called dust cells because we can't avoid eating particulate matter or having it end up in our lungs. Like, as much as our mucociliary escalator is good at its job, it's not perfect. So little bits of dust and other particulate matter do end up in the alveoli of our lungs, and the reason that it doesn't build up constantly and cause cancer or pulmonary obstruction is partially due to cell turnover and partially due to macrophages wandering around in your lungs, hoovering up little bits of dust. So that's an example. Wandering macrophages roam around in connective tissues looking for bad guys. And then remember, interestingly, osteoclasts, which remember are these big multinucleate guys with this ruffly border that dissolve bone. They look like a murder jellyfish, kind of. They're actually derived from monocytes as well, so they come from the same cell line, which is strange because osteoblasts, of course, come from osteoprogenitor cells instead. So there's a different sort of lineage for those two things. All right, lymphocytes. These are supernumerous, so it goes neutrophils, lymphocytes in order of abundance. And part of the reason is that although they all look the same histologically, there are many different kinds of lymphocytes with many different jobs. So you have T cells, B cells, and natural killer cells kind of being the overarching types. But then within each of these categories, you have memory and helper. And then also plasma cells. So this categorization gets very, very complex very quickly with the lymphocytes, and that's one of the things that we're going to have to address more robustly in the lymphatic and immunity chapter rather than here, so I'm just going to gloss over it relatively quickly. So these kind of bop in and out. So they'll wander around the blood for a while, then dip into the tissues, so you'll see them in connective tissue, etc., and then go back into the blood. So they're not confined to any one location. And in fact, they're sort of rambling. So they're kind of like a, somebody who like travels the country all the time. They don't really stay in one place for very long. You know what they say, a rolling lymphocyte gathers no moss. But you see the most of these in lymphoid organs. So in the lymph nodes, in lymphatic tissues in your gut, and guarding your mucous membranes. And we'll talk more about that in chapter 22. So the function of lymphocytes is specific immunity, meaning that they remember the pathogens they encounter so that next time you get infected with one, they will attack it more quickly, and then you never get sick, which is awesome. So this is an FYI slide. I'm not going to read it to you other than to say a differential count is a type of blood test where someone actually goes in and counts the relative abundance of each of your leukocytes and that can be used for diagnostic purposes. So if you saw a lot of eosinophils, for example, you might suspect a parasitic infection or perhaps allergies, just to name one example. So if you're a cytotechnologist or the like, you might spend your work time doing that. Now, I do expect you to know this part. Why did I lose my pen? I do expect you to know the erythropoiesis track for this particular slide, but the rest of this is just for your information, so I won't ask about the rest of them yet, so that we're going to save that for chapter 22. So we didn't really talk about the thymus in the endocrine system chapter because it's this weird crossover gland that does secrete hormones, but it's also involved in the production of white blood cells, and so that it seems more appropriate to discuss that when we talk about the lymphoid tissues. But the basic idea here is that lymphocytes, their development is regulated by the thymus during childhood, and that's because you're acquiring all of your various immunities during childhood. Other white blood cells are activated by colony-stimulating factors. So that's what you're seeing here, here, and here, as well as here. So these are released by lymphocytes and other tissues. So the release of the colony-stimulating factors actually allows fine-tuning of which stem cells are differentiating into which cells, so you can make them according to need, which is nice. Okay, platelets. Platelets, as I mentioned, are derived from a group of cells called megakaryocytes. And megakaryocytes live in the bone marrow, so you don't see them in the blood. They're very, very large. They have multiple nuclei, oftentimes not pictured in this image, of course, but after a while, the multiple nuclei coalesce into one giant nucleus. The platelets themselves do not have a nucleus. So this order of bullet pointing is a little bit misleading. So these guys do have a nucleus. In fact, they have many, and it fuses to form one big one. Here, I'll outline the nucleus of this one. But the platelets themselves do not. They're not actually cells, they're fragments of cells. So the reason that the megakaryocyte needs to have a nucleus is because it has to synthesize the proteins that the platelets are filled with. So platelets themselves are filled with chemicals, and all of these chemicals are involved in facilitating blood clotting during hemostasis, which is the process of stopping bleeding. So platelets have a 9 to 12 day lifespan, and these undergo phagocytosis in the spleen. So just like erythrocytes, which are constantly being produced and circulating and then go to the spleen to die, so too with platelets. Another quick note about platelets is they're sometimes called thrombocytes. That means clotting cell, because a thrombus is a clot. It's a bit of a misnomer, though, because, of course, they're not actually cells. They're fragments of cells. So platelets do a bunch of things. One of those things is release clotting chemicals, so that triggers the conversion of fibrinogen into fibrin, for example, and other clotting factor changes. They also form the initial patch, so if there's a broken blood vessel, they're going to form a patch over the hole, and then that patch is going to initiate the clotting process. And then they also undergo this cool thing called clot contraction. So two familiar proteins to you, actin and myosin, remember? Sliding filament model? Those are also present in blood clots, and they undergo excitation contraction coupling, so myosin cleaving ATP and pulling on actin, to basically pull the clot closer together and close up the hole. So platelets are produced by thrombopoietin, interleukin 6. So this interleukins are another class of chemicals by which white blood cells talk to each other. So inter means between, leukin means white blood cell communication chemical, specifically a protein, and then 6 is it is just the order that it was discovered in. So interleukin 1 was discovered first, interleukin 6 was discovered the 6th. And also multi-colony stimulating factor. Okay, so coming to the end of our discussions of blood, we do need to talk about clotting. And that's because hemostasis, or the process of clotting, is really critical to survival, such that if it's interrupted in any significant way, it's very, very difficult to remain alive. So you've got 3 phases of clotting. The first is the vascular phase. And the vascular phase is followed by the platelet phase. So we'll go through each of these in great detail. I'm just giving you a preview of what is coming down the pike for you. So the vascular phase is mediated by the blood vessels that are broken. The platelet phase, like it sounds like, is mediated by the platelets. And the coagulation phase is the set of chemical reactions and steps by which the clot is matured and comes fully into being. And all of these are important. So the vascular phase lasts about 30 minutes. And I mentioned that this is mediated by blood vessels. So one of the important pieces of this is vasoconstriction. So if your blood vessel is sliced, your blood is supposed to stay in your blood tubes. But if it starts leaking out, like it's going to here, you want to do something in the short term to prevent loss as much as possible until the platelets can arrive and begin to build up. So the best way to do that is just to make the opening really small. So the first step is vascular spasm, which is a step that is mediated by smooth muscle contraction and also, importantly, calcium and calmodulin, because of course smooth muscle contraction is mediated by those 2 things, as well as serotonin. So you need both of those things. The endothelial response to damage, so remember, endothelium are the squamous cells lining the inside of the blood vessel. And these actually contract away from the injury site, so back away from it, and that exposes the underlying collagen. So that might seem counterintuitive, however, collagen exposed is sticky. And so that stickiness is important because that's one of the first steps in getting platelets to land there and collect. So, release of chemical factors happens after that, and that initiates smooth muscle contraction, as I mentioned. There's also mitotic factors to begin to replace cells that were lost, and others for clotting. And then, as I mentioned, the endothelial membranes become sticky as well as the exposed basal lamina, and these help to just make sure that things are sealed shut or bind platelets as much as possible. The platelet phase is very important. So platelets begin to stick to the endothelial cells, the basal lamina, and specifically the collagen fibers of the basal lamina. So ordinarily, the endothelium of a blood vessel should be smooth and slick in order to minimize friction and prevent clots from spontaneously forming inside the vessels. During hemostasis, however, they become sticky so that platelets will stick to them, and you get something called platelet aggregation, which is just the grouping up of platelets on a wound to begin to seal the wound. After the platelets land, they begin to undergo exocytosis, and that releases all of these important chemicals that are needed for clotting, including, notably, calcium and others. So, here's the deal. There is a Goldilocks size of platelet plug. So, the platelet plug formation is a little bit of a positive feedback loop, such that one platelet sticking increases the likelihood of more platelets sticking. So in the short term, that's really good because that's what helps the platelet plug form quickly enough to effectively stop bleeding. However, you don't want that positive feedback loop to cycle out of control, right? So that begs the question, how do you make sure that the platelet plug doesn't grow too large and do something like, say, occlude the blood vessel entirely? Occlude is a word that means blocks or cuts off a passage. Well, prostacyclin is one, and that's just going to inhibit aggregation. So it puts an upper limit on how many of the platelets can get together. White blood cells that arrive at the scene after the damage also basically emit inhibitory compounds that prevent further aggregation. Excuse me, I'm going to cough. I'm going to pause this really quick. Oh boy, that was a loud one. I'm glad I paused it in time. So another feature is plasma enzymes are going to break up ADP near the plug. ADP is one of the clotting factors that we'll talk about. It's on the previous slide. Just know that it's something that platelets release to initiate clotting. And other chemicals. So clot formation also isolates the plug from general circulation. So what that means, if it's not clear, which I don't think the bullet point is especially clear, what that means is it forms a barrier between the platelet plug and other blood cells moving past so that the plug doesn't continue to grow. Okay, the coagulation phase. So coagulation is blood clotting. And I do want to stress, I'm going to move my pen over, coagulation is not the same thing as agglutination. So it's a different set of processes. The only thing they really have in common is making clumps of things, but the things they're making clumps of are quite different. So that's it. I'm just saying that one more time to make sure that you guys don't get those two words mixed up, because if I use them on a test, for example, you need to understand which thing I'm talking about. You know, we're beyond the level where I'll ask you to define those two things specifically, but rather I expect you to be familiar with the definition so that if I use those words in the wording of a question, you're not in the dark as to what I mean. So coagulation is another way to say clotting. So when you see the word coagulation, think platelets, think platelet plug, think fibrin, think fibrinogen. So as the fibrin network grows, blood cells and additional platelets are trapped in the tangle. So fibrin is an insoluble fibrous protein. It's this pink stuff in this picture. So that begs the question, okay, where does fibrin come from? Well, it comes from two possible pathways. Both of them require calcium. So here's another place in the body where calcium is critical for appropriate homeostasis. Again, another piece of the puzzle as to why calcium is regulated so tightly by so many different processes in your body. So we are going to look at the extrinsic and intrinsic pathway. And these are an interrelated set of pathways, really. They're not super different. They have different activation patterns and some different factors. But as you'll see, they have some overlap as well, like most things in physiology do. So let's move on, and we'll talk about each of those independently. So this slide is just comparing and contrasting the platelet phase versus the coagulation phase, specifically as it relates to the relative complexity. So platelet aggregation and the release of chemicals, so ADP, that's adenosine 5-diphosphate, so you can get one of those by taking a phosphate group off ATP, PDGF, which is platelet-derived growth factor, and calcium. So notice there are some things that both the platelet phase and the coagulation phase has in common, and that both the intrinsic and extrinsic pathway have in common, and that particular thing happens to be calcium. So the coagulation phase is the extrinsic and intrinsic pathways combined. So we call these just a series of chemical reactions. So typically it's the conversion of thing A into thing B, and then the conversion of thing B into thing C, etc., so forth, until you eventually turn fibrinogen, which is soluble in plasma, into fibrin, which is not. Depending on the extent of the damage and the state of the plasma proteins, this may begin as soon as 30 seconds after the damage, not always though. And it's the result of a series of steps that involves not 1, not 2, but 12 clotting factors. 12. And that may seem excessive, but if you consider how important stopping blood flow is in the case of hemorrhage, it makes sense that you would have a really robust pathway with lots of backup systems in place. So if thing A doesn't work, maybe thing B will. That's also the putative reason for having an extrinsic and an intrinsic pathway. The argument being, well, if one fails, the other will pick up the slack. So what do you need for this series of steps? Well, calcium, that I keep moaning and shouting about. Also proteins. And these are inactive proteins, like fibrinogen, but other ones as well. We call these proteins. So they've already been synthesized, but they're not actively performing their job yet. They will do so when they are activated. So these typically have a suffix of -ogen on the end of them, like fibrinogen and others. And these are also produced by the liver. So one consequence of liver disease, and including liver disease that's induced by chronic alcohol abuse, is thin blood that is difficult to clot. So one common reason that chronic and very, very heavy alcoholics die, unfortunately, is they end up bleeding out, usually due to gastrointestinal bleeds because of the pressure that liver disease puts on the gastrointestinal vasculature. We'll talk about that in blood vessels, and also because of the lack of clotting proteins in their plasma, basically. So proteins are released from damaged tissues or platelets, and they cooperate with the calcium ions and the liver proteins to sort of do this elaborate, coordinated dance that results in coagulation. So let's take a look at that. So these are all the clotting factors. Note that they are numbered. I'm not going to make you memorize this. This is just a list of them. So the ones you do definitely need to know are fibrinogen and prothrombin, but absent these ones, you don't need to know the specific name or role of these. It's just too complicated for us to get into in this class. So instead, I want you to pay close attention to the fact that the liver is the source of a lot of these. So be nice to your liver. And vitamin K is required for a lot of these as well. So those two requirements must be met for adequate clotting to occur. Interestingly, about half of your vitamin K is made by bacteria in your colon. So they pay rent to live in your colon, and the rent they pay is they say, oh, well, you gave me some food and a warm place to live, so I will give you vitamin K. So it's an example of why you actually need bacteria in your colon, and it's healthy to have some of certain kinds. Okay, so I mentioned that I didn't want you to memorize every single factor in the coagulation pathway, and that's true. So instead, we're going to focus on this slide here. Excuse me, I didn't mean to do that. So one thing I do want to point out is the extrinsic pathway begins in the vessel wall. So specifically, think of extrinsic pathway and associate that with broken vessels. So either by crush injury or by sharp force trauma, but regardless, one of your blood tubes has been released. So you do need to know tissue factor because that's the starter of extrinsic. So the intrinsic pathway begins in the bloodstream, and this is activated in response to factor 12, and that's activated by exposed collagen fibers. So the thing that they have in common is that exposed collagen fibers are part of the vessel wall. So I mentioned that there is a little bit of overlap between the two of them, such that if one fails, another can act. That's the connection that I was having, I was like priming you to make, basically. So extrinsic begins in vessel wall. Tissue factor is released by damaged endothelium, but if your endothelium is damaged, that means probably you're going to have collagen fibers exposed. So typically, the extrinsic and intrinsic pathway, these are active at the same time. So you do need to know tissue factor and factor 12. The other one you need to know is factor X, or 10, because this is the beginning of the common pathway. So what factor 10 does is it unifies the events of the intrinsic and extrinsic pathway such that they all have the same outcome. So what I'm going to do now is fill in what happens here, here, and here to get us to fibrin. I do expect you to know those as well. So from factor X down, and I'll write that here. No from factor X down, no exposed collagen fibers in factor 12 for intrinsic, and no tissue factor and damaged endothelium for extrinsic. So those are the ideas that I need you to keep. Okay, so factor 10, which remember unifies the two pathways, joining them in the common pathway, this is going to activate something called prothrombinase. So remember, I told you guys that a lot of these liver proteins were pre-proteins, meaning they're precursors that are activatable but not active yet. Here's one of them. So prothrombinase is going to convert prothrombin into thrombin. So this is an ACE, meaning it's an enzyme, and it's going to convert pre-thrombin into thrombin. Thrombin is going to convert fibrinogen, which is soluble in plasma, into fibrin, which is not. So the fibrin precipitates out and gets stuck on the platelets, and that's what ends up fully coagulating the clot. Another thing I need to stress is that platelet plug does not equal a clot. It's on the way to a clot, but in order for it to be a fully coagulated clot, you need to have fibrin, and that is the result of the completion of the common pathway. So let's review this one more time, because repetition is our friend when we're learning new things. So this picture down here, before I read you this stuff, is basically just an image that shows you what the subunits of fibrinogen look like, and then thrombin's effect. So thrombin turns fibrinogen with all of these side chains that make it insoluble, and it basically just rips those off and turns them into fibrin. And then fibrin really likes to stick to other fibrins. It's kind of like Legos, where they're the right shape to stick to other Legos, not the right shape to stick to other stuff. And then the fibrin polymer ends up compounding and becoming stabilized. So that's the way this cascade works. So once you've got a mature clot with fibrin, the platelets contract due to actin and myosin interactions. So cross-bridge formation. And that basically draws the edges of the clot and the edges of the wound closer together, which reduces the size of the damaged area. This is an important one. It's basically tightening up that clot and really securing the clot in place so that the healing process can initiate. So I mentioned this previously, but in most cases, both pathways are active due to the type of damage a vessel incurs. So I mentioned this, and I'll say it one more time. That's just to say that if the endothelium is damaged, releasing tissue factor, that means that collagen fibers are going to be exposed, releasing factor 12. So both extrinsic and intrinsic pathways activated simultaneously. Not always the case, but most often the case. So, I mentioned clotting occurs via positive feedback loop, and that's true. However, positive feedback is, of course, sort of regulated by other factors, including white blood cells, etc. The positive feedback loop here is meant to be intense. I got carried away. I'd started to write the F before I wrote the B, and brief. So in general, homeostatic mechanisms are regulated primarily by negative feedback because negative feedback ends itself. Positive feedback will carry on unless it's mediated by some outside force. So positive feedback is therefore reserved for processes that need to be intense and brief. So clotting is one of those. Need to initiate the extrinsic and intrinsic pathway fast and a lot to stop blood flow, but you need to stop doing that when the clot is mature so you don't overclot and block a blood vessel. So clot formation is inhibited by at least 7 agents. We mentioned a couple previously. Also, the calcium ion component. So I pointed out that calcium is required for all 3 pathways, both the extrinsic, the intrinsic, and the common. So if someone had hypocalcemia, you might expect their clotting ability to be impaired as well. I also stressed the importance of vitamin K and mentioned that you get about half of the vitamin K that you possess from intestinal microbes, with the other half coming from your diet. So again, having healthy gut flora and a healthy diet is important for good blood clotting. So let's talk about what can pump the brakes on that positive feedback loop. I was intentionally avoiding saying the names of those things because I wanted to talk about them specifically. So fibrinolysis is one of the ways that clots are managed. And this is both to prevent them from getting too large, but also to get rid of them once they're no longer needed. So let's say that you've had a clot in place and it's sealing up a broken blood vessel, but then underneath that clot, platelet-derived growth factor and other mitotic factors are encouraging mitosis of the endothelial cells underneath. So they're going to knit themselves back together and basically seal up the wall. Once that clot is no longer needed, you don't want it to come loose from the wall of the blood vessel and go traveling as a thrombus. That would be bad. Which means that clot dissolution has to happen in a series of ordered steps so that you don't get an inappropriate traveling thrombus. That would be bad. We call that a thromboembolics, which means a traveling clot. So, another way to say this title is how to avoid a thromboembolics. So, turns out the self-destruct button for a clot is built into the clot as the clot is forming. So, amongst all the fibrin and the red blood cells and the sticky collagen fibers and the myosin and the actin and all of the other components of the clot, there's also plasminogen, which, as I previously mentioned, the suffix -ogen means that it is activatable, but it's not active yet. So as healing proceeds, feedback from healing, so things like growth factors, mitotic factors, etc., begin to convert plasminogen to plasmin. Plasmin digests fibrin strands and basically undoes the clot. So macrophages and other white blood cells are going to come eat up the debris red blood cells that have been trapped in the clot, and the fibrin strands themselves are digested by plasmin. This is going to successfully dissolve the clot such that it goes away piece by piece instead of coming loose in one big chunk and causing damage by blocking blood vessels in the form of a thromboembolus. All right, and that's the end of blood, finally. So I'm going to end this and begin recording the next video for you, and hopefully I will talk to you all soon.